Hello student, today we will discuss a topic for class 12 CBSE book from NCERT book, the leg operon method. But before discussing this, there is a very important thing to know why it is going to be discussed. Okay, we have been studied about central dogma. Central dogma. What is this? It is a special process from which DNA will form DNA itself by the process of replication. Okay, then DNA will form RNA by the process of transcription. And then this RNA will form amino acid, which will form finally polypeptides, and these polypeptides will do some biological function. So we can call it amino acids or proteins. Okay, now by the process of translation. It is the central dogma and it found in all the cells. Okay, now it is going to be very important from DNA, which is a part of nucleus, will form the protein and which will in turn function as a biologically active group. Okay, biological function they will perform. Now, what is going to be important here, their regulation, how they are regulated at every step is very important to know. Now, this is known as your gene expression. What is this? Gene expression, as I have told, from DNA to RNA, then RNA to protein. This DNA is a part of gene, okay, and it will form your protein. This DNA is a part of gene and this gene will form the protein, and it is known as your gene expression. This gene expression need a regulation, okay, and regulation of gene expression is going to be very important, and leg operon model is just an, you can say, an example how the gene expression is regulated. Now we have to see that regulation of gene expression occurs at every level. First, it can be occur at transcriptional level. Very important thing it is. Transcriptional level. Then after transcription, there is a process of sorting, okay, or processing of RNA. Then it can be at processing level. Then after formation of RNA, it comes from nucleus to cytoplasm, means at transport level from nucleus to cytoplasm. Then the last, which is very important, is at translational level, where the RNA will going to form protein at translational. These are the main levels at which this gene expression will be regulated. But environmentally, mentally, physiologically, and biochemically, it is also affected. Chemically, it is also affected. Okay. Now, this gene expression occur in both type of cell, in prokaryotic and eukaryotic. First, we'll discuss in the prokaryotic cell. In the prokaryotic cell. Prokaryotic cell. Actually, in case of prokaryotic cell, the most of the genes are polycystonic. What it is? Polycystonic means one gene can express or can form more than one type of proteins or amino acids. So their regulation, as we have known, there is a transcription unit having a start site which is known as promoter or start site. Okay, we know it and we have been discussed it earlier. This promoter region or a start site is the main region which is going to be recognized by a special type of protein and which will regulate their gene expression. These are arranged in a sequence, and this particular sequence is known as operon, and these are known as operator. These are attached with the proximity with operator region. Now, so finally we come to the lac operon model. Why it is important? Just to know the gene expression regulation in the prokaryotic cell and how it is going to be done. It is done at the transcriptional level, and how it is done, there is a special type of proteins which can be inhibitor or repressor or inducer type, depends upon, and by this action they regulate the gene expression. Now come to the lac operon model. Lac operon model was proposed by Jacob and Monod. In this lac operon model, there is a transcriptional unit, as we know, okay, and there is a promoter region which is your start site. There is an operator region, okay. There is and again a very important region which is I. Don't confuse the I with initiator or inducer. It is always inhibitor in case of lac operon model. And then there are three more important genes, which is Z, Y, and A. Okay. This whole sequence having particular type of or expressing particular type of proteins in association with the operators and promoters are known as operon model according to Jacob and Monod. How it works? Let us see. For example, arrangement of this operator, promoter, and these proteins which are going to be regulating the gene expression and the protein expressive protein. I, promoter, operator, gene Z, Y, and A. This particular sequence are going to form your operon. And why it is known as lac? Because there is a substrate, lactose. Okay. This lactose going to be acted by an enzyme beta galactosidase. And it will form its monomers. And these monomers are galactose and glucose. Now, what is going to be important here? This will work only if it will present. Okay, are you getting it? If it is not present, there are two situations for the cell, prokaryotic cell. If lactose is present, in the cell, then it need enzyme beta galactosidase. If it is absent in the cell, then it will not need. Okay, this is the main fundamental concept of this action or lack operon mode. Okay, now the gene I. What is the function of this gene I? I gene acts as or form a repressor messenger RNA. What it is? Repressor messenger RNA. Okay, it will form repressor, or we can call it inhibitor, because of this word I is present here. Repressor messenger RNA. If there is no lactose in case of in the cell, then it will bind to the operator region. Okay, and when the repressor bind to the operator region, it will be just like that. It will form repressor RNA and it will bind to the operator, and due to this, as it is repress means it is subsiding or suppressing the activity of ZYA, then it will not work. Their work will be terminated. Okay. What's the function of ZYNA? Let us see this. The Z gene, it will express or it will form beta galactosidase. Okay. Then the gene Y functions as gene Y form a protein permease. What is the function of this permease? It will increase the permeability of lactose to the cell. Increase the permeability of lactose to the cell. Okay, I will tell it later. Then what is the function of I? It is transacylation. We have been discussed what's the function of this transacylation. Now, there was a first case when lactose was absent in the cells. This I will form repressor messenger RNA. This messenger RNA will bind to the operator region, and because of this repressor, the ZYA will not express itself, and there will be no formation of beta galactosidase and other proteins. So no. But what in case of what will be if lactose is present? Okay. If lactose is present, then no, just erase this and discuss it. It will form a protein, and let me draw this. It is your protein and it will bind to this, and the whole process is going to stop. But now if lactose is present, it is your lactose will bind to this repressor. Okay, and what is going to form? If I just draw it as this, then it will form this one. Now the function of repressor will stop. Why? Because it will not bind with operator. Very important thing it is, it will not bind to the operator. Now what is going to happen? If it will not bind to operator, then it will just release, and then your Z will form beta galactosidase, Y will form permease, and A will form transacetylase. Always there is presence of little bit permease in the cell, because or so that this lactose can enter inside the prokaryotic cell, and this lactose will able to bind with this repressor messenger RNA protein. Okay, always it is the situation. Just after this, when this lactose bind with the repressor messenger RNA protein, then it will never bind. It is not going to bind with operator. Then Z will form the beta galactosidase. Due to permease, as the Y is going to form permease, the permeability of lactose to the prokaryotic cell increases. So lactose which is present in proximity of the prokaryotic cell will enter inside the cell and will act as the energy source if there is no presence of glucose, and this beta galactosidase will work on that lactose and form galactose and glucose, and the gene A will help in transacetylation. Okay, this was the whole about your lac operon model, and we can see this lac operon model works on the negative feedback model. Negative feedback model. It also work on the positive feedback model, but we will discuss it later. And it was all about this lac operon model. I hope it is going to be at your understanding level, and whatever is that, please write your comments in your comment box so that I can elevate all the elevate my skill, and which will be helpful for your system. Thank you.